Subscribe my channel and press bell icon to get all notification.
Hello students, now let's start the meiosis. So we are going to start with meiosis, and in the meiosis we will start with the meiosis 1. Meiosis 1 or meiosis first. As we already discussed that meiosis process, that complete division by meiosis is completed into the two stages or two phases. One is the meiosis 1 and second is meiosis 2. So we will start with the meiosis 1. In the meiosis 1, we will start the karyokinesis 1. And in karyokinesis, the first phase is the prophase 1. Prophase 1. And here, this prophase is again divided into the subphases, 5 subphases, and the first subphase which we are going to discuss here is the laptoten. So before starting the prophase 1 with the laptoten stage, let's see what happens in these cells during the interphase stage. You know that in the interphase, when the cell prepare itself, cell do all the necessary or required preparation, cell. This is the nuclear membrane which is present in the cell, and chromatin fibers are there. It's get doubled, duplicated. Centrioles get duplicated. They are present here. This is the cell which is present in the interphase, and after interphase, now it is ready to take part into the actual division, which is the meiosis. So it's ready to take part into the meiosis division, and let's start this division. Here, now we will start with the prophase 1, and the first phase of the prophase 1 is the laptoten stage. So we are starting with the laptoten. हम laptoten से start कर रहे हैं. This laptoten is the first subphase of the prophase 1. This laptoten is also known as leptonema. Lapto words means thin, and the nema or nematic word or the T word is related with the thread. So here the thin thread-like structure will form. Let's see what other changes which we can see in the laptoten stage. The cell has a nuclear membrane. Nuclear membrane is intact nuclear membrane. It's not started disappearing. It's present in the intact form. So we can draw it in this way also. We need not to show that it's disappearing. Just make it dark. It's intact nuclear membrane. Centriole is present, it gets duplicated and it's present here. Centriole is present here, and it's not start moving towards the opposite pole. कोई movement start नहीं हुई है centriole की. Nuclear membrane is remain as it is, it's intact, and all the membrane organelle like mitochondria, Golgi apparatus, they all also remain intact. They are also present in the cell. So this is about the cell when it enters into the laptoten, and during laptoten, let's see what changes happens during laptoten. During laptoten, these chromatin fibers. Okay, let's see if we talk about the chromatin fibers here for our convenience. If we take a fixed number of chromosome number, let's erase these. For our convenience, we know that here the cell is the diploid cell. So we are taking a diploid cell to show the meiosis, and in this diploid cell, the number of chromosomes are we can fix so that we can get confused in the later stages of the meiosis. So here we have taken the number of chromosomes 4. In our body, if we talk about the human cell, the number of chromosomes are 46 and they remain into the pair of 23. But that number of 46 chromosomes are not possible to show in this diagram. So we are taking just an example, 44 chromosomes. So here the cells have the 4 number of chromosomes, and these 4 number chromosomes are present in the pair. So we can say 2 pairs are present here. So we have taken a fixed number of chromosomes. When this cell with the fixed 4 chromosomes, with diploid cells with the 4 chromosomes, when it enter into the laptoten stage, these chromatin fibers, here it is in the form of chromatin fibers, these chromatin fibers start condensation. यहाँ पे chromatin fibers condensation show करेंगे, but here one interesting things happen that this condensation is not a regular manner. This condensation happens in the form of at some places, means कहीं कहीं पे condensation होगा और कहीं पे condensation नहीं होगा. So we can see a beaded structure. हमें इस तरीके की structure दिखाई देती है. So in this laptoten stage, we can see a beaded structure. bead-like structure. The condensation happens, but this condensation is not a regular condensation. They get condensed at the regular interval. कुछ जगहों पे होगा, and finally we got a structure which we can say that a beaded structure. So here we can write that chromatin fiber. These chromatin fibers show condensation, or we can say that they show beaded structure. We can see a beaded structure, beaded appearance here. हमें beaded structure दिखाई देगी. And one more important thing that is happen here, that these chromatin fibers, these fibers remain attached with the nuclear membrane. They remain attached with the nuclear membrane by their telomeres. So we can say that they remain attached. देखिए जैसे यहाँ पे हमने attachment show की हुई है, यहाँ पे attachment show किया. They remain attached by their both ends. दोनों एंड से भी हो सकते हैं, but most probably they remain attached with their one end. So here we have seen that these 4 number of chromosomes, we have taken 4 chromosomes, and all these 4 chromatin fibers are now converting into the chromosomes, and they are attached with the nuclear membrane by their ends, by their telomeres. So here this another feature we can write here. This is the characteristic feature, that chromatin fibers, these chromatin fibers attached with nuclear membrane. They remain attached with nuclear membrane. Nuclear membrane के साथ attached रहेंगे. And due to the attachment of this nuclear membrane, we can see a bouquet-like structure. हमें यहाँ पे जो structure दिखाई दे रही है, that structure is just look like a bouquet. So we can call it the bouquet stage. So this stage, this laptonema or laptoten stage, is also known as bouquet stage due to the appearance of these chromatin fibers like a bouquet-like appearance. So these are the changes which happens during the laptoten stage. After laptoten, now cell will move into the next phase, next subphase of the prophase 1. And the next subphase after laptoten, this is the first one. ये हमारी first phase यहाँ पे हो गई है. This is our first phase, subphase laptoten. Now cell will move into the zygotene. This is the cell, and the cell will move into the zygotene stage. Now let's see what happens during the zygotene. Again, the cell nuclear membrane remain intact, no disappearing of nuclear membrane. It will remain intact as it is रहेगी. Centrioles will also present as it is. Centriole यहाँ पे present रहेगा. It is not also starting moving. Centriole की भी movement कोई start नहीं हुई है. Now these chromatin fibers which started condensation here in the laptoten stage, they will condense more and they will look like a chromosome. अभी chromosomes की तरह दिखाई देने शुरू हो जाएंगे. And the attachment we can show, or otherwise if we don't show, that is okay. तो कोई जरूरी नहीं है कि हम इसकी attachment show करें. Now they will look like chromosomes. So we can make the chromosomes. And here one important thing happen is that in the this stage, in the zygotene, let's write the name first, zygotene. This is the zygotene stage, subphase of the prophase. In the zygotene, what happens? The important thing which is happening in this subphase is the pairing of homologous chromosome. So now in the zygotene, the homologous chromosomes will start pairing. First of all, you need to understand what is the homologous. हम क्या किसे बोल रहे हैं homologous chromosomes? In our body, we know that we have 46 chromosomes, and these 46 chromosomes remain in the pair. Means we can say that we have 23 pairs. In these 23 pairs, one chromosomes come from the father and another chromosome come from the mother, and both of these two chromosomes which carry the same alleles of the genes. किसी भी gene के same alleles को carry करते हैं. They are responsible for the same type of genes. These chromosomes are known as homologous chromosomes. So in this stage, in this zygotene stage, the homologous chromosomes start pairing. Homologous chromosomes आपस में pairing बनाएंगे. So here in the starting, in the beginning, हमने कितने number chromosome लिए थे? We have taken 4 chromosomes. हमने 4 chromosomes लिए थे. So here we can see that the 2 pairs will be formed. 4 chromosomes, they will form the 2 pair. तो हमें यहाँ पे 2 pairs नजर आएंगे chromosome का. This is the one pair and this one is the another pair. So this is the one pair and this one is the another pair. So let's see what happens during this zygotene stage. During zygotene, homologous chromosomes show pairing. They get paired. They start making their pairs and they make more condensation भी और ज़्यादा हो गया है and they make pairing, and this connection or this pairing of the homologous chromosomes is known as synapsis. Synapsis is the pairing of these homologous chromosomes. Here, one important thing is that centriole is just as it is not moving towards the pole. Nuclear membrane is as it is. It's not starting degenerating. It is intact. Chromosomes, it may become more condensed, and here homologous chromosomes start pairing, and this pairing of the homologous chromosomes is now known as synapses. Synapses are formed, and these synapses in each chromosome they are making a structure. ये जो दो chromosomes का हमें एक pair नजर आ रहा है, जैसे हमने synapses बोल दिया, but this pair of the chromosomes, these are known as dyad. We can make a word, use word that is dyad. So dyad is forming here, or bivalent condition is forming here. Now let's see here the chromosome which we have drawn here. हमने यहाँ पे जो अपने chromosomes show किए हैं, actually these chromosomes do not look like. इस तरीके से chromosomes show नहीं होते. What happens? Let's draw the structure of the chromosomes separately. अगर हम इन chromosome को enlarge करें, which are present here in this zygotene stage, this is the one chromosome and this one is the another chromosome. These are the two chromosomes, and we know that in the interphase, after in the interphase and in S phase of the interphase, the DNA gets duplicated. Means these chromosomes form their arms. So both of these two chromosomes which are present here in the form of dyad, and they are making the pair homologous chromosomes जो pair बना रहे हैं, इन chromosome में DNA content अब double है. They have two chromatids. Each chromosome has two chromatids. So if we draw the structure, we can draw it like this also, that this is the one arm and this one is the another arm of these chromosome. This is the one arm, one chromatid, and this one is another chromatid. So the structure will look like this, that they have double. There are two arms for each chromosome. This is the structure of one chromosome and this one is the structure of another chromosome. So in this way, we can say that these chromosomes have two arms. They have two chromatids, and if we make it separate, so it means it's not will become comfortable for us. But later on in the next stage, when we show the splitting of these chromatids in the next stages of the meiosis, these chromatids start splitting. इनमें splitting होगी, they will split vertically. They will vertically split it, and by vertically splitting, they form a structure which is known as tetrad. So let's see what happens. यहाँ पे हमने chromosomes को show किया, like if we make the appearance like this one. This is the one chromosome and this is the one chromosome. This is the one chromosome and this one is the another chromosome, and both are two chromatids. Both are two chromatids. When the splitting will happen, these arms will get separated. ये ऊपर की side होगा, same will happen towards the lower side also. At this condition, we can see the four arms. हमें four arms show होंगी, and that structure will अगर हम उसको draw करें, if we make the structure after splitting, so what we will see, हमें किस तरीके की structure दिखाई देगी? We will show the structure of like this. हमें इस तरीके की structure दिखाई देगी. After splitting, when these chromatids get separated, we show the structure like this, and this structure is known as tetrad. This structure is known as tetrad. So later on, the tetrad will also form, and then chromatids vertically gets splitted. The tetrad will form, and here this homologous chromosomes make a pair. A dyad or bivalent is formed. This bivalent or this dyad should be remain with each other. यहाँ पे chromosomes का हमने कहा pairing हो गई है, but this pairing should be remain close to each other, क्योंकि nucleoplasm में present है, they can get separated. दोनों में कुछ separation हो सकता है. इस चीज को avoid करने के लिए, that this pairing do not break up, the one complex is formed between these chromosomes, between these homologous chromosomes, some complex is formed, and this complex or this structure which is forming between these homologous chromosomes is known as synaptonemal complex. That is known as synaptonemal complex. So synaptonemal complex will form to keep these homologous chromosomes together. When these chromosomes remain together, these two chromatids of the nuclear, these two chromatids of the chromosomes, these are the sister chromatids. these chromatids will remain with each other, like this chromosome and this chromosome. Both of these two chromosomes will remain together by this nocturnal complex. And when they remain together, they will make a pairing. They will change their material, genetic material, that we call it crossing over. So when they remain close together, they will do crossing over. The arms of the two non-sister chromatids, like अगर हम इनके arms के बारे में बात करें, these two arms will known as sister chromatid. These two will be sister chromatid. Similarly, these two will be sister chromatid and these two will be sister chromatid. But when we talk about the, if one arm this one and another arm this one, then we call them the non-sister chromatid, because these are the chromatids of the two separate chromosomes. Then we call them non-sister chromatid. So in the later stage, we will see that these non-sister chromatids, they will remain attached with each other. They make a pairing or make a structure by which they do the crossing over. So till now, we have discussed two subphases of the prophase 1, that is the laptoten and the zygotene. Laptoten, beaded structure form in which the condensation happens at some places. That's why beaded structure appears and it remains attached with the nuclear membrane. And in the next phase, which is zygotene, in this phase, the important thing that is happening here is the pairing of homologous chromosomes. So homologous chromosome will make a pair, and after that, in the next stage, we will see that how these homologous chromosomes show crossing over. So keep watching, keep studying, and keep learning.